Hello, I'm Margarita Safonova from Indian Institute of Astrophysics, Bangalore, and this is our team on this project. But first, I would like to talk about the background. What is the extent of Earth's biosphere? Life on Earth is everywhere we look, surviving and thriving in every possible niche, with or without sunlight, water, or oxygen. Some organisms make their own water, take energy from all possible resources, and can hibernate for millions of years. But what are the true boundaries of the Earth biosphere? We know now about underwater and subsurface biosphere, but is there life high up in the atmosphere? Following questions are still unanswered. Vertical limit, does it exist? What is the viability of life upwards? What limits the spread upwards? And can life grow or reproduce in atmosphere or stratosphere, or is it just surviving? From the ground, hundreds of millions of tons of terrestrial dust annually are transported between continents through the air. Just bacterial cells typically exceed 1,000 cells per meter cube over land. Human activity lifts this life into the stratosphere and Earth orbit, with a myriad of aircraft and spacecraft flights, each injecting countless numbers of microbes into atmosphere. In just 2017, there were about 69 million flights around the world, and in just 2020, there were 114 space launches. Orbital space debris of particles smaller than 1 centimeter exceeds tens of millions in numbers. Anthropogenic debris flux exceeds the natural micrometeorite flux by orders of magnitude. It even goes beyond the moon orbit, the so-called empty trash bag objects. This debris, along with its microflora, may eventually settle back into the Earth's atmosphere, but they also may be dispersed by the near-Earth objects of hypervelocity space dust further into outer space. Even on the moon, we left already about 180,000 kilograms of human stuff, 96 bags of human waste. Tardigrades are there, yeast, Drosophila eggs, plant seeds. Moon, Mars, Venus, Titan. In 2015, we have celebrated the end of humanity's initial exploration of the solar system, as every planet has now been visited. And just like the Earth, the Moon is constantly bombarded by space rocks, even more dramatically so due to the absence of retardant atmosphere and due to the low escape velocity. And that poop that we left there may have already been kicked off into the outer space by a meteorite strike. In 2019, the results of multi-species space exposure experiment BioMax concluded that present Mars seems to be habitable for archaea and bacteria. Well, most probably those that we send there ourselves, as 59 various instruments have been launched to Mars by now. Cassini spent about 13 years into Saturn system, had 26 encounters with Titan and a lander on it, 7 close encounters with Enceladus, flying through the geysers of water and organics at 50 kilometers above the surface, and 1 encounter each with the icy moons. Galileo spent 8 years in Jupiter's system, with 11 flybys of Europa through the water organics plumes. Cassini's own discoveries were its demise, said NASA after plunging the Cassini and Galileo into the giant planets. So they were done this to avoid what? Contamination? A bit late? No? Where would the organisms, how would they come if all the assemblies are done in the clean rooms? Well, in the clean rooms, class 100,000 means only 100,000 particles per meter cube are allowed. It is clean compared to the average city air, but it is not sterile. Human traffic in large spacecraft assembly rooms can be more than 50 people at any time, any working day. And humans are the main source of contamination in these environments. We shed 200 million skin cells every hour, and every square inch of our bodies has around 50 million bacteria on it. As NASA engineer in 2015 said, we know there is life on Mars already because we sent it there. These are the pictures of the original assembly rooms of Veneras, and as you see, people don't even wear gloves, no masks. And even in modern rooms, we wear gloves and masks, but half of the face is still open. And very aptly, cartoon was recently published after the Perseverance landed on Mars. And even out of the human space, what about all the pioneers and voyagers, the rocket stages that propelled four spacecraft towards the edges of our solar system and beyond are likely carrying earthly bacteria out in the galaxy now. The upper stages were not required to be sterilized. These bacteria would have been emplaced by the hands and breath of the engineers who built those upper stages. So these microscopic interstellar travelers are now racing out of the solar system. And what about the interplanetary exchange? Today, possibly 1,000 tons of extraterrestrial material rained on our planet. Though fine interplanetary dust dominates this flux, still 25 million meteoroids, micrometeoroids, and other space debris enter Earth's atmosphere every day. Some of it is impact ejecta from the Moon, Mars, Venus, Mercury, and some of it is terrestrial, returns of impact ejecta from the Earth, debris from orbit and spacecraft, unburned rocket fuel, even human excreta, as shows this picture of the dump of the 150 pounds of water and urine from the Space Shuttle Discovery in 2019. So present day low Earth orbit anthropogenic debris flux exceeds natural, possibly at all sizes. And spaceflight experiments have shown that with minimum shielding, a number of types of bacteria, archaea, and viruses can survive at least a short-term, weeks to month exposure to space in low Earth orbit. Multiple studies have shown that life can survive the three stages of traveling from one planet to another: initial ejection after the, say, meteorite strike, the journey through space between planets, and the impact on a new planet or a moon. It was estimated that microbes and spores can drift intact in space for up to 1 million years, and, if embedded deep in the rock, even after 25 million years in space, a substantial number of spores could survive. And we know now that all terrestrial planets were habitable early on. Mars had surface liquid water very early, large global magnetic field, and was a warm greenhouse. Venus could even have been more habitable than Earth, with surface water, moderate temperatures, and even a magnetic field. Since its origin, the Earth biosphere was in constant interaction with atmosphere, hydrosphere, or cryosphere, and space. Saturnian moons, Enceladus and Titan, Jovian moons, plus Neptune's moon Triton are strong candidates to host oceans. If a sufficiently large asteroid or comet strikes Titan, for example, it would puncture the icy crust and expose a vast reservoir of the subsurface liquid ammonia water mixture, ejecting it into interplanetary space. And recent experiments have shown that microbes can make their own microhabitats and thrive even in the cold. Flammable ice in the Sea of Japan inside the flammable ice, the researchers found microscopic spheroids of oil and water in frozen gas and ice created by microbes making their own ecosystems. Authors concluded that providing they have ice and a little heat, all those frigid cold planets at the edge of every planetary system could host tiny microhabitats, with microbes building their own death stars and making their own tiny little atmospheres and ecosystems. What about astrobiology in our stratosphere? Earth's stratospheric conditions are very similar to the surface of Mars or even to some exoplanets. Studies in the stratosphere can be a test run for looking for life, in other words. For example, Martian surface parameters are nearly the same as Earth at 30 kilometers altitude. What could be the source of the biomass in the stratosphere? Well, it can be arid topsoil, marine sea spray, dust storms, volcanic activity, and recently a lot of human activity: landfills, agriculture, airspace traffic, pollution, space debris. And it is known now that the residency time of airborne species, at least in the lower atmosphere, is up to 5 years. Microbial habitation in troposphere is known and well documented. It is known that bacteria continue to grow at a height of at least 3 kilometers in supercooled cloud droplets, but stratospheric reports are not without controversy. We know perfectly well how the IDP look because of the collection by the space mission Stardust and Dawn. However, previous stratospheric isolates, some people, including Vikramant Singh, Narlikar, and Wainwright, they claim what they collected by their balloon collectors was the biological entities. They claim that these are collection of, well, bacterial cells, and this something what they say has even a proboscis. However, we know that proper stratospheric samples from balloons and airplanes contain Chondritis IDP and megaspores. I put here several of the pictures of the megaspores to show that some of them may even resemble that proboscis. So ET claims were published in this International Journal of Astrobiology in 2010 that these bacteria-like particles were found to express some characteristic there of non-terrestrial, but they don't say which. They say that cells have been found to be viable, but they didn't mean how. What did I mean by viable? And they lack an important characteristic of terrestrial microorganisms, which is the ability to proliferate in normal cultural conditions. But then how do they think they proliferate in space? The clusters of non-culturable cells may have been fossilized, carbonized, deactivated, and subsequently degraded following prolonged exposure to cosmic rays and UV radiation. But then if stratosphere degraded them, how do they live in the conditions of space, which are much worse than that? Some studies have also reported that some microorganisms found in stratosphere are successfully isolated and cultured. They contradict themselves. Either bacteria cannot be cultured, and this means that they are extraterrestrial, or they can be cultured, but if we can culture something, that it means that they are terrestrial. In 2003, Narlikar and Toll reported the results of the balloon flight collection of air samples from about 40 kilometers altitude as detection of extraterrestrial microorganisms raining constantly on Earth from space. They found clumps of kiss-shaped sub-micron-sized particles falling on Earth at a rate of 3 tons per day. In addition, some terrestrial species were discovered. But their balloons were launched from India in TFR balloon facility in Hyderabad. These are zero-pressure balloons capable of carrying 1 ton of payload, reaching 50 kilometers and floating for hours. For that, the payload releases the ballast, which is can be up to 10% of the payload mass to boost the balloon and the payload to the higher altitudes. Ballast that is used in TFR balloon facility consists of tiny iron balls. Subsequently, the payload sucks in the air through the micropole filters for collection. So here is on the left is the results of the Narlikar and et al. Collection of tiny balls, which they claim to be characteristics of clusters of coccoids, microfossils, and so on. And on the right, I am showing the picture of the ballast iron balls. So these are very tiny, that they can be up to few maybe 100 microns, maybe less than 100 microns. So it is quite possible then when this ballast was released and the payload on their balloon sucked it in, or sucked some parts of these tiny balls. When in 2015, Wainwright reported high-altitude balloon flight during the Perseid meteor shower, where they claimed to have discovered weird biological entities, including a titanium shell bacterium and a diatom frustule, presumably coming down to Earth embedded in cometary micrometeorites. They, however, gave little detail of a payload and the sampling chambers, and all the conditions under which the payload was assembled and later disassembled. At least for the diatoms, reports are available showing their widespread in the upper atmosphere. In addition, only a broken part of a diatom frustule was found in 2000 in the Wainwright flight, and these are ubiquitous in any dusty environment, a so-called diatomized Earth. In fact, their launch was from a place near Ellesmere Port, Cheshire, UK, and the company Cheshire Pump Co. Ltd., located in Ellesmere Port, Cheshire, UK, is one of the leading UK suppliers of the diatomous. In 2013, our group at Indian Institute of Astrophysics started the high-altitude balloon program. These are some of our photographs from stratosphere. We usually have two cameras, one looking up at the health of the balloons and one looking at the horizon. Some of our objectives are essentially UV follow-ups of transients. We have to go above ozone layer, and we are a UV group. And testing and verifying the operation of our payloads on balloon flights with the intention of eventually placing them on small satellites. And developing flying low-cost scientific payloads on balloon-borne platforms, also developing instruments that can operate on a range of near-space platforms, including CubeSats, mini-satellites, and space missions. Our balloon experience has been invaluable as training and verification for our current space activities. What do we need to fly balloons? We need lots of permissions. Airport Authority of India, local airports around Bangalore, some other Delhi people like Ministry of Defense. Balloons we procure from Pune. They are not very expensive. 1.2 kg balloon is about 2000 rupees. We need, of course, parachutes. We need gas. We started with helium, which is safe. However, helium, one cylinder costs about 20,000 rupees, which is very expensive. Hydrogen, not so safe, but with proper training, we use it now, hydrogen, because it's much cheaper. Then, of course, we need ropes, electronics, and payload. What payload constitutes sensors. We always fly environmental sensors, temperature, pressure, humidity. We have attitude sensors, cameras. We have trackers, GSM, GPS, radio trackers. Batteries, of course, we use lithium polymer because they work in low temperatures. And 2 days before launch, we calculate balloon flight path using flight path prediction software. Sometimes we use our own codes. Some of the instruments that we have developed: flight termination unit, data logger, radio trackers, attitude sensor, even star sensor. Radio tracking, we use ham frequency. We have our own ham radio and our call sign is this. And we put our base station, the high elevation Omni antenna. In addition, we are developing the balloon-borne payload system called SAMPLE, Stratospheric Altitude Microbiology Probe for Life Existence. Like, what are the two boundaries of the biosphere? What is the vertical limit? Can life grow or reproduce in the atmosphere? So this, we hope to collect dust samples from stratosphere and return them back in an uncontaminated manner to further study for the possible biological isolates. It should contain power supply, controller, we use Arduino RPi, environmental sensors, of course, GPS trackers, and radio transmitter. This was our first prototype, however, only on a drawing board, payload box, and this was supposed to rotate. However, we ran into difficulty of coating the collecting area. So one of our students suggested, why don't we use the spent DVD drives from the old computers? We can coat this with aluminium foil and then coat with agar, use Arduino control, and this particular prototype was tested in a -80 degrees chamber for 2 hours for survival. It passed. So when we took it out, it worked. And when we put it back again, 2 hours operational, we tested it for 4 times open-close cycle. It passed. So what about the coating? Usually in stratospheric flights or even the space flight, they use polyurethane foam or they use aerogel or zerogel, but these are very expensive. So we try to use agar-agar. So in one of students from IIC department, he made a test because usually agar concentration is 2% for culturing. So when we coated this tray with 2% agar and put it in -80 chamber, which is usual temperature in the stratosphere, where we pass through -80, then it was flowing. When it was brought back to +20, it melted and flew away with the water. So amount of water lost was measured, and inversely he correlated it with the extent of maintained agar architecture. So finally he found that 10% agar is the most stable after the freeze-thaw cycle. In addition, in Pushina, I got a suggestion that if agar is based not on pure water, agar solution, but on water plus glycerin, then at glycerin concentration of 40%, even after freezing to -70, there was no crystallized formation, which is also very important. So we are contemplating this also. The only problem that glycerin and agar already has a lot of organics, so maybe that will contaminate slightly, confuse our subsequent determination. So what is the operation that we plan? Before coating the collecting trays with the medium, we would clean them. The necessity is not for introducing the biota, because even if it is killed later, sterilized, since it may confuse the later identification. So that is why the collection trays will be placed in the sonic bath filled with isopropanol solution at our laboratory. Then we'll place trays into sterile bags and deliver it to the IIC department for sterile coating with agar. We'll return and store it in 100 class box in the lab. An hour before integration with the balloon payload, all surfaces will be wiped with the IP solution. We also have to go above 23-25 kilometers because, as this paper reported, that at about up to 20 kilometers they collected chondritis porous aggregates which contained silicon-rich contaminants from volcano eruptions. So we plan to program our single-board computer to extend the tray above 23 kilometers and retract it once it is crossed again on the way down. Upon the recovery of the payload, we'll deliver it to the class 100,000 clean room in our lab, where the collecting trays will be removed from the payload box. After their surfaces will be wiped with the IP solution, they'll be placed in a sterile bag to deliver to the examination lab. We will also use two controls. One tray, absolutely the same way prepared, we'll keep in the lab, and one tray will fly, but we will not extend it. So what are the investigation methods? Physical methods may include scanning electron microscope, but however morphology is tricky, as I have shown before. So mass spectrometry is possible because, for example, Earth terrestrial dust will rarely have iridium or osmium in its composition. Isotopic content for biological examination. For example, if there is higher ratio of C12 to C13, that would suggest biological origin of carbon, or high deuterium ratio, or nickel to ferrum, like in metallic meteorites, will suggest extraterrestrial origin. Life on Earth also uses light isotopes of carbon and nitrogen. Biological methods. Many Earth organisms cannot be cultured. Culturing is very nutrient environmental specific, and spores is especially difficult to culture, so we will not use culturing. Something what is called metagenomics. Metagenomics is a genomics technique without the need for isolation and lab cultivation, culturing of individual species. It relies on the extensive existing DNA libraries. However, even metagenomics may not be possible, as an organism may be not even DNA RNA based. We know that there is something proposed shadow biosphere, like for example, desert varnish. It is not based on DNA RNA, and it's possible that it is alive. Or the sequence may not be available in the library. After all, we are aware only of less than 10% of all the living species on Earth. In the recent years, something came up which is called nanolive. With nanolive imaging, a low-power laser beam gets phase shifted when sent through samples. This phase shift is then used to generate 3D images of refractive indices of the observed samples. The result is quantitative 3D cell tomography in vitro, without any invasion or destruction or sample preparation, staining, with a resolution of down to 90 to 75 nanometers. This is the movie from the nanolive preparation of the cell mitosis. This is video of our launch in a speed up. This was a atmospheric launch. That is why it is not in the night. We are astronomers, but it was atmospheric launch, so it is in the day. So this is some references of our heritage and experiences. And for the we have our lab, MJK Menon laboratory clean rooms, from class 100 laminar flow tables and class 1000 clean rooms to class 10,000 clean rooms and class 100,000. See, this is our assembly and alignment of lunar ultraviolet cosmic imager Lucy that we have created and we designed and assembled, and it is now stored in class 100 box. This is that Lucy. Thank you.